In this video, we'll move to the next level of structure within protein molecules. And it's the chemical nature of the natural polypeptide that puts constraints on the shapes that it can take in 3 dimensions. This means that proteins will have a defined secondary structure, and there are 3 types of secondary structure: the beta sheet, the alpha helix, and loops. So let's see how these elements come about, but we'll first see how the constraints on the polypeptide will give rise to the different secondary structure elements. So recall that proteins are polymers of amino acids, and these polymers come about from condensation reactions that are catalyzed by the translation machinery in the cell. And that leads to an unbranched polymer of amino acid residues that are linked by amide bonds. And in proteins, these amide bonds are called peptide bonds. In the early days of protein biochemistry, measurements of the dimensions of the peptide bond led to a surprising finding. The C-alpha atom has the expected tetrahedral geometry, and the carbonyl carbon has angles corresponding to a trigonal geometry, suggesting that the peptide bond is a double bond. But the peptide bond, the bond between the carbonyl carbon and the amino nitrogen, measured 1.32 angstroms, and therefore it is shorter than a typical carbon to nitrogen single bond, but longer than a carbon to nitrogen double bond. This unusual bond length is due to the partial double bonded character of the peptide bond because of resonance, as shown here in this chemical equilibrium. An important consequence is that the peptide bond is planar. The nitrogen and hydrogen of the amino group and the carbon and oxygen of the carbonyl all lie in the same plane. A second important consequence is that the peptide bond cannot rotate freely, unlike normal single bonds. So the peptide bond can be in only one of 2 conformations, cis or trans. And I'm talking about the dihedral angle, which states the position of the 2 C-alpha atoms that are linked through that peptide bond. In the trans conformation shown here at the top, the 2 C-alpha atoms are on opposite sides of the peptide bond, whereas in the cis conformation, they're on the same side of the peptide bond. But as you see here, when they're on the same side of the peptide bond, the 2 side chains tend to clash, and that means that the cis conformation is unfavored. So almost all peptide bonds are in the trans conformations, with one exception, and that's the peptide bonds that are N-terminal to a proline. Recall that prolines are cyclic amino groups, and that means that the steric clash is similar in both the cis and trans conformations. And since the trans conformation is now as bad as the cis one, it is much more likely for a cis peptide bond to occur before a proline. And note that it is the peptide bond on the N-terminal side of prolines that is affected. So now we know that the peptide bond is fixed, but the other 2 dihedral angles of the backbone on either side of each C-alpha carbon are free to rotate. Each residue therefore has 2 such dihedral angles, one named phi between N and the alpha carbon, and the other named psi between the alpha and the carbonyl carbons. An easy way to remember the order is that it goes alphabetically. In proteins, N always comes before C, and phi or phi comes before psi or psi. Peptide bonds are also named. They're named omega. Omega is either 0 or 180 degrees for cis or trans bonds, respectively. Let's see how the phi and psi angles are measured. To measure the phi angle, you look down the length of the nitrogen to C-alpha bond from its most N-terminal end. And then you measure the angle between the 2 carbonyl carbons. Similarly, to measure the psi angle, you look down the length of the C-alpha to carbonyl carbon from its N-terminal end, and then you measure the angle between the 2 nitrogens. Each residue therefore has 2 such dihedral angles. So this isoleucine has dihedral angles phi of minus 138 degrees and psi of 132 degrees. Here I've plotted the dihedral angle values for this isoleucine on a phi and psi plot. Now, a famous Indian structural biologist, G.N. Ramachandran, realized that not all dihedral angle values are allowed, and there are some steric clashes. So using molecular models, he tested all possible values of dihedral angles, and then he realized that there are only 3 regions that contain allowed values of psi and phi angles for normal natural proteins. And this plot that you now see is called the Ramachandran plot. Let's take a closer look at how the plot was built. This animation illustrates a full rotation around the phi bond of a valine residue. The corresponding dihedral angles are indicated in the Ramachandran plot on the top left. Steric clashes are illustrated by cylinders that you see appearing at some angles. Note how the allowed angles here within the blue region of the plot minimize those clashes. And we can do the same process doing a full rotation around the psi bond. Ramachandran looked at every single possible positions to build his plot. We can see now again the 3 regions of allowable phi and psi angle values on the Ramachandran plot. Now, to form the 2 familiar regular secondary structures, alpha helices and beta sheets, we need several residues in series within one given region of this Ramachandran plot. So let's see a specific example. For an alpha helix, we need multiple residues in a row to lie within the phi and psi region shown here in green. And here is a cartoon representation of such an alpha helix. This cartoon representation, which was invented by Jane Richardson, shows a helical ribbon for helices. Now, natural L-amino acids form right-handed helices. So imagine if you're going up a staircase that's shaped like this helical ribbon. The railing that's on the outside should be in your right hand if it's a right-handed helix. Now let's look underneath this ribbon at the backbone atoms. You can see that the backbone atoms, when they're in this phi-psi region of the Ramachandran plot, can form hydrogen bonds with their polar groups, so that the carbonyl of the residue N will hydrogen bond to the amino of the residue N+4, as highlighted here in yellow. Side chains, which are illustrated here as spheres, will then point outwards and slightly downwards on an alpha helix towards its N-terminus. Now, the periodicity of an alpha helix is that it contains 3.6 residues per turn, and that will lead to important patterns in the primary structure or sequence of a protein. And I've illustrated here with an example from ATP synthase. So on this helix, you can see the yellow residues are the hydrophobic residues, and they all fall on one face of the helix. And if you count them in the primary structure, they're all about 3 or 4 residues apart. And that allows this particular helix to form a hydrophobic core by packing against another helix, which I'm now showing here in blue. Here's a quick brain teaser for you. Why do we sometimes say that prolines break helices? Can you think of why that is? There'll be a question about this at the end of the video. Let's now turn to beta sheets. Now, multiple residues in a series that fall within the blue region that's illustrated here on the Ramachandran plot can form a beta strand. And then you need multiple beta strands to hydrogen bond together to form a sheet. Here is a 4-stranded beta sheet in cartoon representation. Each strand is illustrated by an arrow going from the N to the C terminus. Sheets can contain parallel strands, anti-parallel strands, or a mixture. As you can see here, this beta sheet is mixed. Anti-parallel strands often come from adjacent parts of the primary structure, and then they would be linked by a short loop. But adjacent strands can also come from a whole other part of the same polypeptide, or from another protein altogether. Again, let's look at the backbone structure behind these cartoons. In a single beta strand, the backbone is in an extended conformation. And then the hydrogen bonding of backbone amino and carbonyl groups between adjacent strands is what forms a beta sheet. Looking at the edge of a beta sheet, you can see how the side chains extend perpendicularly to the plane containing the hydrogen bonds. The side chains within one beta strand alternate above and below the plane. So in beta sheets, we often see an alternating pattern in amino acid properties. For example, alternating aliphatic and polar residues could form a sheet with one hydrophobic side and one polar side.
What about the last region on the Ramachandran plot?
Yes, so this combination of phi-psi angles is also allowed, but it cannot be repeated for many residues, unlike the other ones. And so this region will correspond to loop regions. But be careful. For loops, any allowable phi-psi angles are okay. So you'll find loop residues in all three regions of the Ramachandran plot. So remember that to obtain an alpha helix, you need a repeating sequence of amino acids that are all fall within the green region of the Ramachandran plot, and it would be the same for the beta sheets in the blue region of the Ramachandran plot. So a residue that's in the alpha region, that doesn't mean it's in a helix. It could also be in a loop. So to get a regular secondary structure, you need phi-psi angles to be within a similar region, all in a series. Loops and turns will connect adjacent secondary structure elements. Our first example here is two strands of an anti-parallel beta sheet, which are connected by a small loop. And then a second example is a helix followed by a loop followed by a strand. Most loops will fall at the surface of proteins and contain a lot of polar and small residues, and they'll also form the shapes of active sites because they have a lot of conformational flexibility to really shape themselves around the ligands. Remember we talked about two special amino acids, proline and glycine, both of which have a lot of interesting conformational properties. Glycine being very flexible and proline being restricted but being able to form cis peptide bonds. And so proline and glycine are often found within loop regions of proteins. Let's wrap up by looking at alcohol dehydrogenase, an enzyme important in energy metabolism. It contains a mixture of alpha helices, beta sheets, and loops. You can see that accordingly, its residues are distributed in all three regions of the Ramachandran plot. So we've now seen how the backbone of a protein can be arranged into defined elements, the alpha helices, beta sheets, and loops. This level of organization is the secondary structure.